Good afternoon, everybody. We'll go ahead and get started. It's really terrific pleasure to be with you and welcome Andrea Armani to the Institute of Optics and giving us some Andrea here around Halloween is perfect because given the amount of things that she gets done, the amount of different directions, there's got to be some sort of supernatural force that's active. I don't know exactly what it is, but Andrea started her academic life at the University of Chicago, where she got her degree in physics, and then moved on to Caltech, where she had worked towards a PhD and got a PhD in applied physics with a minor in biology. Working with Kerry Valhalla on ultra-sensitive label-free sensors. And that's really where I first learned about her work with these silicon toroid resonators that she was developing there. She stayed on at Caltech as a postdoc before moving to her independent faculty position at USC, where she very quickly established an incredibly productive and creative group working on important biological problems, applying tools of sensing and other detection technologies in ways that could solve these problems that really weren't accessible using other techniques. As she rose through the academic ranks, she acquired a number of awards along the way. I'll just list a couple of these. She got a National Institutes of Health Director's New Innovator Award in 2010. These are incredibly difficult grants to get and really highlight particular creativity at an early stage. She was named to Popular Science's Brilliant 10 in 2014. And many others that I won't list here. She became the Ray Armani Chair, named Chair in Engineering and Materials Science at USC, as well as Professor of Chemical Engineering, Materials Science, Electrical Engineering, Mechanical Engineering, and Biomedical Engineering. So I think that's a familiar with that problem of having many possible committees you can serve on.
Yes.
But also speaks to the diversity of what she does. Recently, she moved to the Ellison Institute of Technology, where she is now Senior Director for Physical Sciences and Engineering, as well as retaining her academic position at USC. So very excited to have her here and looking forward to her telling us about nonlinear optical imaging.
Thank you very much, Ben. Wow. You just set a really high bar for me.
It is.
Okay. So as Ben just mentioned, I did recently just move to the Ellison Institute. So I'm disclosing I'm now at the Ellison Institute, which is a company that's affiliated with USC. If you have any questions, I can answer them. And that's really all there is. All my grad students who get their degrees at USC moved with me. And since my grad students really are the ones that do all the work, I always like to acknowledge them at the beginning. As was mentioned, my team is very interdisciplinary. So I have PhD students that are getting degrees in a wide range of fields. So I kind of color-coded them to a certain extent. I run out of different ways to try to explain interdisciplinary. But you're going to see kind of throughout the talk how chemistry and chemical engineering and material science and EE all come together to support the research. Or at least I hope you see that and that comes through. Cross my fingers. So the nice thing about being at the Ellison Institute where I am now is that the technology development or the engineering part serves as the foundation for basic science, which then goes right into the clinic. And I remember one of the first times I met with Ben, he was talking about how he was working on research and working directly with clinicians, which is an amazing place to be. And it's one of the amazing things about Rochester as well, that you can directly interface and interact with clinicians. So if you're students and you're working on a collaborative project, don't just hand your widget over to the clinicians, talk with them. A lot of times they have problems that they don't realize that you have a solution for. So make sure that you actually talk with your clinical partners. Don't just view them as a black box partner. Go follow them around in the clinic for a day if they'll let you. They'll learn a lot. But where my group lives is at the bottom of this. We're really doing the technology development part. Although I do send my students into the clinic. Again, I think it's invaluable. It's really great experience. So today I'm going to talk briefly about bioelectric fields. In other words, give all of you a kind of a really fast crash course in what bioelectric fields are. Then go through some of the new nonlinear optical imaging agents that we've made and explain exactly what that is. And then a few examples of how we're actually using them. This can be interactive. In other words, if you have a question, you should feel free to interrupt me. This doesn't need to be like a one-way conversation. So this can go both ways. So bioelectric fields. All of you are probably very familiar with bioelectric fields, even if you don't realize it. So an example of a bioelectric field are neurons, right? They generate electric fields in their biological system. Hence bioelectric field. Usually when we try to characterize neural activity, we use things called calcium indicator dyes. In other words, we're just looking at light and changes in light as an indirect readout of the bioelectric field activity. You can also use things like neural probes or metal probes and like directly jab. That's a whole other category. But if you're going to use an optical approach, you're going to typically use something like a voltage indicator dye, which will allow you to visualize. The downside of using a voltage indicator dye, or one of them, is that you can see the activity, but you can't actually control the activity. In other words, if you're trying to perform an experiment, you always want to be able to control it, right? You want to be able to say, okay, if I perturb it in this way, how does that change the activity? And now if I perturb it in this other different way, how does that change the activity? Right? You want to be able to do hypothesis-driven research. You don't want to be a bystander in your own measurement. Right? You always want to engage. In this kind of, if you take a step back, you can think about your bioelectric system or your neural network as a network. And all of you are optical engineers. In other words, you're engineers. And the idea of a bioelectric field network, you should bring back the idea of a network system. And the electrical engineering side of me, when I started thinking about network, that brought to mind wireless networks and wireless communication networks. That's what a neural network is. And when you want to characterize a network, you start thinking about networks in terms of nodes and in terms of stability of the network, robustness of the network. More importantly, what happens if one of those nodes drops out or maybe just becomes intermittent or maybe actually increases in strength, right? How does that affect the entire rest of that network? And if all you can do is record the strength of the bioelectric field, you can't really ask any of those questions. And that's a huge area of neuroscience that can't be studied. Any of those questions, well, the idea of a node falling out or a node increasing in strength, those are the exact analogs to Alzheimer's disease and epilepsy. So they directly map to diseases. So that's why it's important to be able to have a model system. The other huge benefit is we already have all the mathematical models. This is some really nice people in communications theory. So, yay. So when we start trying to think about, characterizing a communications network, right? We already have the wireless version. We now just need to think, how are we going to adapt this to the wired neural network version? And are there probes that we can actually use to allow us to both read out the bioelectric activity as well as write the bioelectric activity? In other words, controllably turn on and off neurons without actually interfering with the entire network. And even though I've really focused in on neurons, bioelectric fields aren't limited to just neurons, right? Your heart cells, the reason why they actually cause your heart to contract, your muscle cells to contract, that's also a bioelectric field. It's why when you think someone might be having a heart attack and you have to resuscitate them, it's huge electric field pulses to resuscitate your heart. So this isn't necessarily like a toy neural problem, although no one would say the brain is a toy, but it's not limited to just neurons. Like all the cells in your body are controlled through bioelectric. So it's a very interesting, very big picture problem. So we wanted to develop a probe that would allow us to both read and write bioelectric fields. It also wouldn't interfere with the system. And it also could be used to map out entire tissue. Keep in mind when I say we, this isn't like just my group, like everybody in the world. There's an entire initiative called the Brain Initiative in the US. There's also one in India. There's one in Europe. There's one in China. There's one in Australia. Everybody wants to do this. This is like a big picture goal. Hence, there are lots of people trying to do this. The three most common ways being attempted. The first is an electrical probe. So this is like a simple picture, but you can take an electrical probe, like a metal electrical probe with an array of wires. Maybe the electrical probe is kind of flexible, maybe it's made out of plastic, maybe made out of silicon nitride, but an electrical probe of some ilk that allows you to read out electrical activity as well as inject electrons into the tissue. Keep in mind, you're now forcing something into tissue, which intrinsically is going to damage it, right? It doesn't really matter how flexible you make it, you're still going to cause some point of sight damage. Also, you can only control or read out the neurons that your probe touches, right? So there's kind of a limit surface area. The huge advantage of these is they're really, really fast. So you're able to get very fast signals, so the electrical probe can actually keep up with your neuron firing frequency. So that's a huge advantage that the electrical probe has over all the other methods. Then you have optogenetic methods, which I'm assuming everybody's heard of optogenetics. Oh, okay. So if you learn one thing today, learn about optogenetics. This is something all of you should know about. So optogenetics, you genetically manipulate a cell to be responsive to a specific wavelength of light, typically blue. And then because your cell is now responsive to blue light, you're able to control its electrical activity, turn it on or off. That's the bio part of it. So that's the genetic part of it. Now, the opto part of it, there are a whole host of complementary optical methods that have been developed, like tapered optical fiber probes that can be inserted into the brain, free space optical fiber probes, silicon nitride tapered probes, tapered probe arrays that can then expose and conduct blue light to then excite those genetically engineered cells to then do those control measurements. Right, and they've done this genetic engineering of animals. So they have like little mice that can run around with optically controllable brains, and then they can study motor function, as an example, which is really cool. One downside, you have to genetically engineer the mice, right? So if you have to genetically engineer something, then you don't necessarily know if the system that you're studying is the actual system because you're genetically engineering it. But optogenetics is a huge field. All of the students here, you're in optics, you should know about optogenetics. It's a thing that you should learn about. Because there's a lot of technology development that can still happen in optogenetics, and you could end up in a job with that. So patch clamp. The last is patch clamp. This is probably the oldest method. Patch clamps are basically glass pipette tips that you put like a really light vacuum on and you suck part of the cell membrane up into the patch clamp, and then you measure the conductance across the end of your glass capillary. All right, so if this sounds like it's an art, it's because it's an art. All right, imagine trying to just pull a little, like pinch a little bit of a cell membrane into the end of a glass capillary and then measure the voltage change across it. So you can do it at like a single cell, as long as you're really, really patient. And you're only looking at a single cell, not like a tissue slice or an animal. But patch clamps, again, because it's an electrical approach, you can get really, really fast signal. So you can keep up with the speed of whatever it is you're trying to measure. So pros and cons of all of this, right? I like optogenetics. I'm an optics person. But the electrical approach, the electrical probe by far right now outstripping optogenetics approaches. But they're on different slopes in terms of technology development. So we decided to take an alternate path to all of those. So all those previous methods are tethered. In other words, they all rely on a cable to actually get the signal out. We decided to take an approach much more like those calcium indicator dyes, where you can just monitor the fluorophore and record changes in the optical intensity of the fluorophore and determine the electrical activity. And so we used one molecule that acted as our recorder molecule, so it would tell us the bioelectric field activity. And then we were going to use another molecule, another chemical compound, that would act as a modulator. So it would actually control the bioelectric field intensity. So in electrical terms, right, we have a recorder and a modulator. And then obviously we would want these two molecules to be co-located. So we would just stick them together with an insulator so that they didn't talk to each other. Right, so we could avoid crosstalk. And these two molecules would be independently controlled because they would be controlled by two separate wavelengths of light. So one of them is a 2-photon material, so it's controlled in the near IR, and one of them is invisible, controlled molecule, so it's controlled in the blue. So they're super simple. Everybody gets how this works? So since we want this thing to actually directly interface with neurons, that means that it needs to actually go into the cell membrane. So we designed it to be a little bit hydrophobic so that it would self-localize into the membrane because the cell membrane is a hydrophobic environment. We also engineered it to be about 4 nanometers long so that it would nestle nicely inside the cell membrane. So that worked out really well. And we did this by putting a little tiny hydrophobic tail on it. So the tail goes in the membrane. I'm making all this sound really easy. Should be really glad my PhD student isn't here. Because I'm just like, yeah, we just stuck that tail on there. Right. It's fine. She's gonna like defend in 2 months right now. She's like, yeah, I did that. Yeah, it's great. I have a job. I don't care. That's the best thing. Like 2 months for you to defend, like you just jump there. Yeah.
Quick question. If you go back to your assignment diagram, for your modulator part, you want to modulate the effects of the ion channel. So just being the membrane, how do you make sure it's close enough to the ion channel to actually be able to modulate? Yeah.
Yeah. So this is a cartoon.
Yeah.
This is a better cartoon. So really the whole thing actually inserts into the membrane. Keep in mind, we don't just have like one in there. We make this using solution chemistry. These aren't nanofabricated, they're actual organic molecules. So when we like dope a cell with them, to try to take more EE type words, we're using like 50 micromolar, 100 micromolar type concentrations. So there are many dimolecules per cell. So sure, some of them are just going to end up in the cell membrane. Some of them may end up attached to a receptor on the top surface of a cell and do absolutely nothing. And some of them are going to actually end up in a channel where it's not. And all of that is a really important point. And the reason why we don't have really high background noise is something I'll talk about in a minute. So this is the actual molecular structure. So the blue part is the modulator part. The red part is the light emitting part. And we have 2 modulator arms on there simply to increase the efficiency of the modulator. The recorder part, having that ratio of 1 to 2 was kind of a good balance. The little black chain, so that little black chain, that acts as our insulator. We actually looked at a couple different insulators. This is the first generation version. We've now switched to a PEG insulator, which is a little bit more hydrophilic. We went a little strong in our first design. The nice thing about making your own molecule is you can also change your own molecule and swap it out a little bit. So the read mechanism. So how this entire mechanism works, so how we're actually able to detect bioelectric field is normally when our molecule is all happy and in a good environment, we expose it to light, it is excited to a higher energy state, it decays, then it emits light, single one photon fluorescence. In the presence of an electric field, we end up with a non-radiative decay. Obviously, if we were just looking at a single fluorophore, it would be binary, right? It would be either one or nothing. Now, because we have a population of molecules, we now get a population of states. And so we're able to get a range of intensity readouts. So it really relies on this not being a single molecule readout, having many of them reading many spots on the surface of the cell. So not work in a single molecule configuration. Also, the absorption and emission wavelengths are dependent on the absorption states of on the energy levels and absorption of the specific molecular structure. So we did some DFT modeling to optimize the chemical structure and to optimize the energy gaps. One question I've gotten a lot, so I just want to address it, is speed. So calcium indicator dyes, the reason why I keep going back to them is because they're like the classic dye. Calcium indicator dyes usually have responses in like the milli to microsecond timescales. Ours are closer to nano to picosecond timescales, so it's faster, which means we'll get faster readout. So we'll actually be able to detect our neural signal. So getting to your question about why I'm not worried about dye that has bound to incorrect portions on the cell. So most conventional dyes, they emit light all the time. So you're able to detect the fluorescence when it's in low concentration, when it's in a medium concentration, and when it gets to a high concentration and it starts to the material that we made actually has the inverse behavior. So when it's in a very low concentration, it actually is non-emissive. And only when it is in a very high concentration and the molecule either starts interacting with itself, in other words, you have two molecules and they are interacting with each other, or it starts interacting with the environment around, and its vibrational and rotational motion is restrictive, will it emit light? Which means if we have dye molecules that are just kind of freely bound on the surface of the cell, but are still able to rotate, they won't emit light, so they don't generate any signal at all. We don't detect them. So this reduces like random background noise from our molecule. This behavior is not something we discovered. I want to be really clear. This behavior is a known behavior called aggregation-induced emission. There are a lot of organic molecules that undergo aggregation-induced emission. We just snagged one of them, but it's a thing. Normally, molecules like fluorescent undergo aggregation-caused quenching. Just the count. So for anyone who's super excited about synthetic chemistry, this is how we made it. There are really only two super important points to note here. The first is that it's kind of modular. All right. So the second component that forms the actual light emitting block, and then that light emitting block is directly attached to the modulator block via that second reaction. Now we did some magic with our chem draw. But the alkane chain is shown there with like just a single carbon bond, parentheses 8, because it was an 8-unit carbon chain. But we can actually change. So it's very much a Lego-like organic chemistry scheme. So it's easy to swap what that insulating chain is, to swap what that light emitting group is. In other words, if we wanted to, instead of having an oxygen on the end, have a sulfur group on the end, we could do that. So you might say, why would you want to have a sulfur group on the end? Well, it's a little bit more on the drawing. It also is a little bit more compatible with bacteria cultures. So you can really play around without being forced to go all the way back to the beginning on the synthesis. So I describe it as like Lego chemistry, simply because it's very flexible. Now, after we confirmed the light emission behavior, we also needed to confirm the photoconductivity. So the NAI, which is our photoconducting group, was one of the original photoconductors invented by Xerox. So Xerox, before most of you were born. This is like an old school molecule. It's why it's really important to go back and read literature when you're going to do something new, because sometimes you don't necessarily need to actually do something new. You can just take something old and do something new with it. So a photoconductor is a molecule that conducts or converts photons to electrons, right? And that's exactly what we wanted to do. We wanted to convert photons to electrons and then have them get injected into a cell. So we just took NAI, and then we needed to figure out how to conjugate it and make it biocompatible. But the NAI molecule and the synthesis of NAI was very well worked out and very well optimized by a big company. So we took all of their chemistry and just adapted it. And so it wasn't that big of a shock that it actually worked, but you always want to make sure it worked. More importantly, it also worked in once it was conjugated to our readout molecule. And more importantly, our complete conjugated molecule also exhibited the AIE behavior that we wanted it to at reasonable concentrations. And one question I got over lunch was, what is your biggest concern about biocompatibility? You might notice that the fraction on the bottom there is THF, which is tetrahydrofuran. So that's the solvent it's in. THF is not a biocompatible solvent. A biocompatible solvent would be something like water or buffer, maybe salt water. So we did this measurement in THF. We showed it worked in THF. And then we quickly had to start making some modifications to our molecule to actually make it work in other solvents that wouldn't kill every single cell that it came into contact with. In the process of doing that, we also discovered that the material demonstrated something called sabatochromicism, which means that the light that it emits depends on the pH of the solvent that it's in. So this is just an example of the molecule in a whole host of different solvents and the emission wavelength changing. And then there's a theory that predicts exactly what it should emit and showing that it works. So this is an interesting thing to note because it means that the molecule can actually pick out the pH of its environment. So if anybody in here does something called FLIM, which is fluorescence lifetime imaging, if the wavelength is changing, then that also means that the fluorescence lifetime should be changing. And it means that the same molecule could actually pick out different pHs. Before I get asked the question, because I was already asked during lunch, no, we have not tried. Yes, we are going to try. No, we have not tried yet. I'm 1%.
So another thing that's interesting about that is that your band structure is also changing with the solvent. It looks like, I think that's the DMSO spectrum, that you've got some extra stuff going on there. Why is that?
I don't know. Really good question. I'm not sure. Yeah, there is some extra stuff. There are two possible reasons. Impurities, because we made it, and sometimes there are residues from the prior step that can get stuck in there and not filtered out, right? Because they were slightly in there, but they're much more present here. And this data, we basically had to take every single fraction that we had that was kind of purified to take this data. So it could be impurities. And then the last thing that we really wanted to check was to make sure that our claim that each one of those molecules was actually individually addressable was actually true. Claiming that you have two molecules that you can control independently and that you actually don't have crosstalk between them when they're only separated by a few nanometers is important. So we confirmed that we could actually excite one at 350 and get the predicted emission wavelength, excite the other one at 390, and they didn't overlap with each other. And we got the same spectra as if they were two separate molecules. And then we also took a 2-photon absorption spectra of just the TE molecule. So the big question, does it actually work? Right, all this was just making a material. So I'm going to skip over a lot of the biology sample preparation, but if you have questions, I can ask. So in collaboration with Brown, we decided to test it in something called a mini brain, also a neural organoid, or a brain microtissue, or neural microtissue. They have lots of different names. The key thing about this type of microtissue is it has all the different types of cells that a normal brain has, except it has been recapitulated into a culture plate, and you can use them with a normal confocal microscope. Also, because it's in a culture plate, you can infuse dye into them. You usually seed them at a few thousand cells and then let them grow for several days, and you can image them at discrete day intervals. As long as you image them early enough, then they actually vascularize and they have random as well as coordinated spikes like an actual brain. So this is two sets of data. The top row is one set, the bottom row is a second set. I always like to show the data that didn't work, especially when I'm giving a seminar, maybe not like a conference. So the top row is the data that was like our first attempt, and we didn't really know what we were doing, but it at least showed that like we had hope. So it was a solution that was unfiltered. So there was both good and bad material in there. And you can see some important things like there was clearly clumping on the surface at like 2 hours, at like 6 hours after injection of the dye. It looked like, there was really nothing on there. And then at 8 hours, the neural organoid had begun to get like smaller and smaller and smaller. And by 10 hours, the organoid was dead. So that was not, the fact it was getting brighter, that was not a positive sign. It was getting smaller. So the dye itself was getting closer together. So more green was not good in that case. So we revisited our formulation. We also put in a little bit of detergent to try to increase the ability of the dye to actually be injected into the solvent or into the buffer around the neural organoid. This allowed us to use a lower concentration of dye. So we went from 50 micromolar at a starting of 37. We then went to 13 microliter to, and I'm putting unknown there because our collaborators don't know how to calculate. Right? I'm working on this. Yeah. They haven't sent us the numbers yet, but they're going to. I know the number they sent me was wrong. I'll put it that way. I know it was wrong. So that's also why their names are not up there. So as you can see going across, the light, the intensity is very, very uniform across. The organoid does start to begin to shrink and get smaller here and here to here. So there is clearly some cell death happening, which isn't great, which we're still going to continue to work on at this point. But importantly, this is a confocal image. And the fact that the dye is actually getting into the middle of the neural organoids, is actually able to infuse through this microtissue to get in the middle, was really significant, and we were really excited to see that, because we were actually kind of concerned that like it might just stick around the edge and we would just get like a ring of dye around the outside of our neural organoid. And it didn't completely kill it at day one. So that was positive. So this, we actually co-incubated while the organoids. This was a co-incubation process. That's all I have on our preliminary tests. We did actually try to do like a like imaging showing that we can detect blinking and firing. We can detect that the neurons are firing, but they look like they're twinkling. That's the only adjective I have. So the intensity fluctuations of our dye is faster than the resolution of the camera that they have. So we're now getting a high-speed camera and putting it on their confocal system. But our dye looks like it twinkles because it's twinkling really, really fast. Whereas their calcium indicator is more like a slow modulation. So we can't, other than saying that yes, our dye is responding because it's twinkling, we can't actually put any numbers on it. Pictures are pretty.
Those are really pretty pictures. Just a quick question on, since your dye, right, is just inserted into the membrane, but cells are pretty dynamic with the membrane composition, they go in the cell, come back out. How does that maybe present an artifact for twinkling and blinking, right? Is it just coming out of the focal plane, or is it coming back in because of the cycling of the membrane, or is it actually safely there? And
that is a great question, and that's one of so the current measurement that we're doing, the actual experimental design is we're going to do a simultaneous co-excitation with an electrical probe to basically force a spike and then show that we detect the spike at the same time with our dye, so that they better be synced, right? Because if they're not synced, then that means what we think we're detecting is not what we're actually detecting. So that's like the ultimate control measurement. So we're going to do that. But as far as like cycling in and out, you are 100% correct. They could be cycling in and out. Ideally, if it cycles into the cell, it should then be able to freely rotate. So it should turn off. It may not. That's a question we don't know the answer to.
How deeply have you seen the dye penetrate to date, and you expect it to be the same in brain tissue or in the retina specifically?
So we haven't looked in the retina. I know where you're going with this. We've only looked in organoid type structures.
Are they expected to have the same properties as more or less?
More or less.
There's an IOM in the retina that's, prevents things from getting through into it. That might be prohibitive, I don't know.
Yeah, so these are like probably 300 to 500 microns, I would guess. I mean, so far we haven't had like the biggest problem we had was initially getting them in, and then when we started using like a really low concentration of detergent, that problem went away. We're also in parallel looking at doing like a micelle or vesicle mechanism to also improve delivery. I'm a big fan of doing developing two methods in parallel, and we weren't sure if the detergent method was going to work, so we also started the micelle method. And we're looking at both active and passive formulation of the dye into So the last example I have that I have real data on, not just picture, is actually using just the light emitting part to try to understand surface receptors on the surface of cells. It really takes advantage of that AIE property of the dye. So in biology, particularly in cancer cells, the type of receptors and the distribution of receptors on the surface of your cells can play a role in a lot of regulatory pathways. There's a really famous surface receptor, at least in breast cancer, known as HER2. There are a couple different types of subtypes of breast cancer. One of them is HER2 positive breast cancer, which means that you actually have the HER2 receptor on your cell. Importantly, it isn't just the presence or absence of the HER2 receptor on your cell that's important. It's actually the distribution of the receptor on the cell that's important. And this often gets overlooked because typically people talk about developing a diagnostic to detect HER2, just the receptor. But there are therapeutics that actually target disrupting the receptor distribution. In other words, breaking up the clustering of HER2. So being able to detect both the presence or absence, but also the distribution is a relevant question. So that's what we were curious about, was if we could detect the presence of HER2, but also detect the distribution of HER2 on the surface. That being said, just to give you a big picture, HER2 is part of like a family of receptors that are present in breast cancer. There are a lot of receptors that play a role in breast cancer. The other pair that are important is the EGFR receptor. So the HER2 EGFR clustering behavior is also important. This time we focus on the HER2 dimerization / clustering. So there are many ways of trying to study the HER2 clusterization, obviously, because it was discovered, right? So somebody has to have imaged it before. The most common way is to do single molecule microscopy, some flavor of single molecule microscopy. Who knows about single molecule microscopy? I'm getting more hints on that one. Okay. Either that or you just don't answer my questions. Okay. I'm asking for like a hand. I'm not going to call on you. Okay, just be clear. Just because you raise your hand, I won't call on you. So there are lots of flavors of single molecule microscopy. The one that was actually used to discover the HER2 clustering was FRET, which means you have one fluorophore here, you have another fluorophore here. The emission wavelength from this fluorophore overlaps with the absorption wavelength from this fluorophore, and then you can tell how close they are to each other. That's how the HER2 clustering was discovered. Since that time, there are lots of other ways that it's been detected and monitored and confirmed. But all of these are really common in a research setting, not so common in a clinic setting. So it's not necessarily great for trying to do a high throughput screen to determine distribution of HER2 on a cell surface. So we wanted to see if we could use our TPE molecule in a high throughput setting to avoid the need for single molecule imaging. So we took our TPE molecule, our AIE molecule, conjugated it to an antibody that was specific to HER2. And the idea was that if you have a cell surface that has a low concentration of HER2, you'll see a low signal. If you have a surface that has a high concentration of HER2, you'll see a and a high concentration of clustering, you'll see a really bright signal. And the spatial resolution based on our modeling was between about 2 to 6 nanometers, so they'd have to be pretty tight clusters for this to actually work. Oh yeah, and I had my student take a picture of solution versus aggregates. So it's like really, really bright if it aggregates. That's not like water solution, like there's actually AIE molecule in it. So the conjugation process took about 2 hours. We used a monoclonal HER2 antibody that we bought commercially. Before someone asked, yes, we started exploring peptides. We've actually designed and had 2 peptides made that we're now exploring. trying to anticipate the questions I normally get. So we first confirmed the antibody conjugation worked using mass spec. We saw the mass spec shift like we expected based on the increase in mass from the antibody. We think that we had about 4 dimolecules per antibody on average. Right? That means that some have more, some have less. And also the emission wavelength, the absorption emission wavelengths didn't change significantly. We looked at 2 cell types, HER2 positive and a HER2 negative cell type. HER2 negative is our control. Even a HER2 negative, like all cells have some HER2. So HER2 negative really just means HER2 low. There's also right now in the field of breast cancer, like a whole debate about is it actually like HER2 positive or should it be HER2 high and HER2 medium? Like it's wading into that debate. It's not fun at all. So there's the HER2 negative / low one. You can see a little bit of a signal because there is a little bit of HER2 even on the HER2 negative cell line. But on the HER2 positive one, you can very clearly see a signal. And these images were taken on a 10x objective microscope, right? Like, oh, sorry, 20x objective microscope. But wide field, this is not like a tiled image. It is single stride. In other words, very lazy. That's it. So then we decided to test colocalization to make sure that our TPE dye was actually attaching to the HER2 site. So colocalization with a Texas red. So the top is our dye, the TPE dye with the Texas red, and then merged. And wherever there is green, there's also red. So our dye is sticking where it's supposed to stick. The bottom, we did the same thing, but we used fluorescent instead of CPE. And we got the same result. And just to confirm, like truly same result, you can calculate something called the Pearson correlation coefficient, which means you go and do a pixel by pixel analysis and look to see if everywhere there is red, there's green, and then you plot. And if everywhere there is red, there is green, it should be like a straight line. And ours was actually looked better than the fluorescein one. Just saying. You know, okay, so then we decided to see if it actually works. So there's a therapeutic called trastuzumab, also called Herceptin. As a side note, if you see a drug and it ends in NAD, that means it's an antibody-based therapeutic. Shorthand for your intellectual enlightenment. So trastuzumab is a therapeutic that's designed to split up HER2 dimerization. That's like its goal in life. So we decided to see if it actually worked, if that was actually the mechanism of action. Because there were some other theories that maybe it was binding at the HER2 site, but doing something else farther downstream. So we did this by plating some SKBR3 cells, some of our HER2 positive cells, running a range of incubation times, running a range of concentrations, and then looking to see if there's actually a change in fluorescence intensity. So when we did this with fluorescein, like a conventional fluorescent dye, we saw no change, right? But you wouldn't expect for there to be a change because the fluorescence isn't changing, right? The receptors are just moving around on the surface, but there isn't actually a change in the number of receptors. When we did it with our dye, we actually saw a slight decrease in intensity, and the decrease in intensity both depended on the concentration of Herceptin and on the duration of exposure. So it double depended. So that was great. Also, I should add, all of this data was taken on something called an operetta, which is a high throughput, high content imaging machine. So it was taken not manually. So it was taken in an automated fashion and analyzed automated fashion. So let me grab you right now. I'd like to thank all of you for your attention. And as I mentioned, we're now beginning to move into peptides. We're doing therapeutic screen just for curiosity, and we also are heading into more flume studies and phase imaging studies. So thank you, Ben, and I'm happy to take any questions.
Can you comment on the right function of your technology and its applicability potentially for vision restoration?
So the application in vision restoration, so it's triggered in the blue. So it theoretically, yes, but obviously would need to be combined with something like a blue source.
And how bright would that source be? You know, that's one of the challenges of current technology.
Yeah.
It takes so much light, and you're exposing damaged retinas to an awful lot of light.
Yes.
Yeah, right.
Yeah.
Do you have any notion of how sensitive?
So right now, the blue light that we're using is we're just using a blue LED. We aren't using like a high power blue pulse laser or anything like that.
So are you doing any nonlinear optics here or doing 2-photon?
So 2-photon imaging for the read, but the write is 1-photon.
Okay.
The write is very boring. I'm sorry. I'll try to step it up. I'll get to work on that.
So with the Herceptin work, even at a high concentration of the antibody, you still have a pretty high intensity. Why is that?
It has a really high quantum yield. And then also it has the added advantage of having very, very low background noise. So like I told, so the student that led this is a chemical engineer. And when she started the project, pre-COVID, I explained to her that there are two ways to tackle imaging challenges, right? You can either try to increase the signal or decrease the noise. Because that's really all you have, right? Signal and noise. And this project really focused on decreasing the noise, even though it seems to be focusing on the signal. But because the molecule doesn't generate light unless its motion is restricted, the noise, the background noise, goes away. Because all of the like unbound, freely diffusing molecules are non-emissive. So then the only molecules emitting light are the ones that are bound to the HER2, and specifically the HER2 dimers. So then you end up with very specific signal. So it's somewhat counterintuitive, but it's really the whole project was to decrease the noise.
And I guess a follow-up that I don't know whether it's a question or a comment, but since you can do this time resolved, you should also be able to study receptor dimerization. This technique.
Which is why we want to go to peptides. It's the exact motivation. So for those who don't know, peptides are smaller, so they're a little bit, they can move a little better. So there.
Do you think there's any possibility like, because you brought up like neurological disease like Alzheimer's that have like disrupted neurological function, activity, and then modulating that with your molecule could. So what do you think it's possible embedding in like a photoconductive polymer sort of array that you can then maybe onto a whole brain or something, and maybe do like, since this looked like you were exciting at blue versus like you said like 800, right?
Mm-hmm.
If you kind of shifted them, their band structures a bit, and you could do 2-photon excitation and like some brain stimulation. Do you think this could potentially be an application of that today, or?
So I want to say yes, but that is so far out. Perhaps I'm just a really big realist. Getting things into the brain, really hard. Getting things like drugs into the brain and getting them to stick. Just like promising things and not delivering. And I'm not sure how feasible it would be to be able to do something like that in a very reliable manner. And in order to control neural activity in that way, your dose is going to be really important. And for this dye, the dose is going to be dependent on the concentration of the dye at the cell. So you would need to have incredible precision over the delivery of the dye at the cell, and it would need to not move. All right, and cells die and replicate. So that's a huge problem with biological systems. That cells die and replicate. And then suddenly you have no dye in your cell because your cell decided to die. I mean, it's just, yeah. Right? I mean, cells shouldn't die. They shouldn't. They should just stay there forever.
That's the problem with more complex biology too.
Right. So I'm being half sarcastic, but it's something engineers, like you can't forget. Like biological systems are fundamentally unstable systems. And you can't treat them like they're stable systems. Because they are ever evolving and ever changing. And you can do an experiment on one day and then do it the next day and get a different result. And it's not because of your experiment, it's because your systems change. And so it's a mindset that you always need to remember. Like biological systems are ever evolving and ever changing. Oh no, I promise.
You mentioned that you had to add a hydrophobic tail at the beginning, but you switched to something more like why did you have to be that?
Yep, so we added a hydrophobic tail, but like a really hydrophobic tail. Because we went overboard. And so we switched to something still hydrophobic, but not as hydrophobic, right? Like they're hydrophilic and hydrophobic, that they're bookends, and then there's things in the middle. So we just went like a little bit less hydrophobic. We should.
When you mean like you went over, what did you see?
Oh, like it just we couldn't get it into biocompatible solvent. It just forms chunks. It was very pretty, but it formed chunks. Like it was non-useful.
So for the when you're looking at the brain or at your brain cells, you're doing this confocally, which means you're taking point data and then making parcels. If you have the advantage of being able to be time sensitive for the signal triggerings, have you thought a way of also being spatially sensitive such that you can analyze more than one node at the time on your network? Or
So we haven't yet. Right now, we've been focused on basically just being able to optimize the imaging conditions.
okay,
Because like any new dye, right, you first have to optimize all the imaging conditions just to get a picture.
yeah, I mean,
Yeah.
given that they're short nodal it's wild to be able to nonetheless get
A picture.
blinking. Yeah.
So the kind of our next series of measurements, just to give you an idea, we're going to go through, just like we can selectively target for HER2 on a cancer cell, we're going to go through and selectively target for a subset of the cell types in the mini brains. So instead of just labeling the whole mini brain, we're going to go through and selectively label subsets. So then we can monitor the firing of subtypes of cells in the mini brain, which I think will be interesting, instead of monitoring all the firing. And then we're also going to go through, this is obviously after we do the control measurement to make sure that we're seeing what we think we're seeing. That's step one. Then we're also going to go through and build a hyperbaric chamber and start looking at how firing is dependent on environments, like oxygen-rich environment, oxygen-depleted environment, things like that.
Okay.
I have a feeling I'm about to get parts. Yeah,
You mentioned you're upgrading the camera to higher
I'm not my collaborator, it's because
higher frame rate imaging. I was wondering, like how much of an improvement do you need and to like get the time resolved information? And then looking beyond that, like what are the temporal resolution limitations of your imaging modality?
So our dye in terms of a fast. The camera they're gonna get is gonna be more in the nanosecond timescale. So it's they're looking to keep up with the neurons, not with our dye.
Okay.
Which is fair.
Well, I think given the time,
I think
we'll
it's
end there.
Cut off.
Yeah, let's thank Andrea